Hello everyone, Dr. Khatami is here. Welcome back to another anatomy lesson. Today we're starting chapter 9 all about skeletal muscle tissue. Muscle tissue is a primary tissue that is specialized for contraction. That means it helps us move, whether it's walking, blinking, pumping blood, or even moving food through the digestive system. We also learned that there are three types of muscle tissue. We have skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. So they are different in their location and also different in their structure and a little bit in their function, like skeletal muscle, which is attached to the skeleton, to the bones. It acts voluntary and move bones. Cardiac muscle tissue is only going to be found in the heart wall and it's going to be controlled involuntary and it's going to contract and pump the blood out of the heart. And the smooth muscle tissue, which is also involuntary, it would be found in the wall of internal organs, like in the wall of the small intestine, in the wall of the stomach, in the wall of the bladder. In this chapter, we are going to focus specifically on a skeletal muscle tissue. We'll learn how it looks, how it works, and how it helps us move. Let's talk about what it actually does in the body. Skeletal muscle has several important functions. Let's go through them one by one. Skeletal muscle tissue can produce the body movement, which is the most obvious job of a skeletal muscle, is to help us move. When these muscles contract, they pull on tendons, which then pull on bones, and that's how movement happens. This includes simple motions like reaching out to grab something or taking a deep breath, and also more complex activities like dancing, swimming, or playing piano. So that is the obvious job of a skeletal muscle tissue. Produce the body movement. Next, it maintain posture and body position. What does that mean, maintain posture and body position? Skeletal muscles are constantly working to keep us upright and stable. Even when you're just sitting still or standing, your muscles are active, like holding your head up, keeping your back straight, and stabilizing your joints. It might not feel like much, but your muscles are doing a lot behind the scenes. Next function of the skeletal muscle tissue is to support our soft tissues. Skeletal muscle also form protective layers in the body. It forms the abdominal wall and also the pelvic floor. These muscles support our internal organs like your intestine, bladder, uterus in female, and keep them in place. Next, we have guard body entrances and exits. Some skeletal muscles form sphincters. Sphincters are circular muscles that control opening in the body, which would be found in digestive system, urinary tracts like your mouth, anus, and urethra. These muscles give us voluntary control over important activities like swallowing, urinating, and defecating. Next, it also maintains the body temperature. That is very interesting. When your muscle contract, they generate heat. In fact, muscle activity is one of the main ways your body stays warm. So when you're cold and start to shiver, that's your skeletal muscles working hard to raise your body temperature. So skeletal muscle tissue is going to maintain the body temperature, which we need in our body for normal functioning. And next, the skeletal muscle tissue also can help us to store nutrients. But how? You know, skeletal muscles contain a large amount of contractile proteins. During times of nutritional deficiency or starvation, when the body doesn't get enough nutrients, especially proteins and energy from food, skeletal muscles break break down some of their proteins into amino acids, which then the amino acids would be released into the blood and then would be used by other parts of the body for energy or for repair. So as you can see, our skeletal muscle tissue are amazing structure. They can help us to move, they can help us to maintain posture and body position. Even when not moving or just sitting still or standing, they can protect and support our soft tissue and internal organs, like they're going to be found in our abdominal cavity and pelvic cavity. They can act as a sphincter to control the opening of the digestive and urinary tract. They can maintain our body temperature, which is important to remember. And also, they're going to be important in store nutrients as they can be break down into the amino acids in during the nutritional deficiency and starvation. So, as you can see, skeletal muscle does way more than just help us move. It supports, it protects, it controls, warms us up, and even feeds us when necessary. Let's talk about how the skeletal muscle are connected to our bones. Most skeletal muscles run from one bone to another, so we can actually define two muscle attachment. So as you can see, muscle would be attached to the bone through the tendon, which we know tendon is a dense connective tissue. So we are going to define two muscle attachment. One bone or one muscle attachment will move, but another muscle attachment remain fixed. So we can call one muscle attachment as origin and another as insertion. So we have origin and insertion. The origin is the less movable attachment, and insertion would be the more movable attachment. What does that mean? When muscle contracts and make movement, so it's gonna move the bone over another bone, right? Okay, so when muscle contracts, that is the insertion moves toward the origin. So look at this picture. Here you can see the brachialis muscle, which would be found in the arm region. And then we can define origin and insertion or the fixed muscle attachment, which would be our arm bone or humerus, and another muscle attachment site, which is the forearm bones. So as you can see, insertion is moving toward the origin, means that the forearm bones or the forearm would move toward the arm. So that is the way that we can bend our elbow when brachialis muscle is contracting. So make sure you know the definition of origin and insertion, which means origin is the fixed part or the less movable attachment, and insertion is the more movable attachment. When the muscle contracts, that is origin moving toward, that is insertion moving toward the origin to make the movement. Now let's talk about the structure of a skeletal muscle. If you look at the skeletal muscle which are connected to our bones, like the muscles over the shoulder and the muscles in arm, forearm, or every muscle attached to the bone in our body, like your biceps muscle, your hamstring muscle, your quadriceps femoris muscle. The muscle, the skeletal muscle, isn't just made up of muscle cells. It's actually a complex organ. It's made up of muscle tissue, yes, but it also contains connective tissue to hold everything together, blood vessels to supply oxygen and nutrients, and nerves to control muscle contraction. So it's really a full team of structures working together. Now let's break it down step by step from large to small. So if we cut the muscle and look at the cross section of the muscle, you will see that each muscle is actually composed of many muscle fascicles. So each of these small circles inside a muscle would be a muscle fascicle, which the muscle fascicle is the bundle of muscle fiber. So then here you can see we have the muscle fascicle, which then inside of a muscle fascicle is made up of many muscle fibers, like a group of straws held together by plastic wrap. Then inside of each um, fascicle, we have muscle fibers, which actually muscle fibers are muscle cells. So we have muscle as an organ, then each muscle as an organ is made up of muscle fascicle, like this. Then each muscle fascicle is made up of many muscle fibers or muscle cells, like this. Look at these long and skinny cells, like threads. Okay, so if you cut the muscle cell and look at the cross section of muscle cell, you will see inside each muscle fiber or inside each muscle cell, we find myofibrils, which myofibrils that you can see like a little dots inside of the muscle cell. These are long cylindrical structures that run lengths of the cell. So then here we have inside of muscle fiber, we have myofibril, which then we will see that each myofibril have many myofilaments, which myofilaments are tiny protein filaments made of actin and myosin, which these proteins are responsible for contraction. So let's review it one more time. We have a whole muscle as an organ. Then each whole muscle as an organ is made up of the muscle fascicle. And then each muscle fascicle is made up of many muscle fiber or muscle cells. Each muscle cell contains myofibrils, and each myofibril contain myofilaments. And one more thing, you know, and I said the muscle is an organ, is a complex organ. It's not just made up of the muscle tissue, it also has connective tissue, it has blood vessels, it has nerves, right? Let's see where the connective tissue would be found or going to be involved in making the skeletal muscle organ. So the connective tissue can make a covering for each part of the skeletal muscle. We have epimysium, which is the dense, regular connective tissue. which is a strong and flexible layer of connective tissue which gonna wrap around the entire muscle, and we call this as epimysium. Just remember, epi means above, and myo or means muscle. So epimysium means like the connective tissue above or over the entire muscle. Then the epimysium actually protects the muscle from friction and keeps it separated from surrounding tissues and organs. It also provides pathway for nerves and blood vessels to enter the muscle. So it's not just wrapping. It also can be a gateway. Then we have another connective tissue which is gonna cover each muscle fascicle this time. And we call this as perimysium. So again, peri means around, and we also have this term myo or myso, which means muscle. So then the perimysium, which is the middle layer connective tissue, it surrounds each muscle fascicle. Like the epimysium, it is also made up of dense connective tissue, and it helps protect and support the muscle fibers inside each muscle fascicle. Then finally, we have endomysium, which endomysium is the innermost layer connective tissue covering each individual muscle fiber or muscle cell, which this time is a thin layer of areolar connective tissue. The endomysium, this areolar connective tissue, contains capillaries, contains nerve fibers, which then they deliver nutrients and signals directly to each muscle fiber. So it's super important for keeping each cell healthy and active. Okay, I know that it gets so messy here. So let me just add this. Endo means inside, right? Okay, so let's see. The structure of a skeletal muscle, which we discussed about it, we know it's an organ like your biceps muscle, like your triceps muscle, because it's made up of muscle tissue. It also contain connective tissue, which like epimysium, endomysium, perimysium. It also has blood vessels to send them nutrients, oxygen. It also has nerves to send them signals from our brain and nervous system. And then we learned that each muscle as an organ is composed of the muscle fascicle, which then each muscle fascicle is a bundle of muscle fibers. Make sure you know the muscle fiber is the same as muscle cells, and then each muscle fascicle we have muscle cells, and then each muscle cell is composed of myofibrils, these cylindrical structures, which then each myofibril is composed of myofilaments, which is the protein filaments. It's going to be important in muscle contraction. And we also covered the connective tissue sheath in the skeletal muscle. We learned about the epimysium, perimysium, and endomysium. So the epimysium is the outermost layer surround the entire muscle. This is a tissue, is a dense irregular connective tissue which protect the muscle from friction and also separate the muscle from surrounding tissue and organ, provide a pathway for nerve blood vessel. We also learned about the perimysium is also a dense connective tissue going to be found around each muscle fascicle. And then we learned about the endomysium is the innermost layer. It is made up of areolar connective tissue full of capillaries and nerve fibers. It will surround each individual muscle cell or muscle fiber. Now it's time to draw. Let's draw and label a skeletal muscle structure, muscle organ, fascicle, fiber, myofibril, and also connective tissue surrounding each. Are you ready? Let's do that together. Let's start with the whole muscle, the muscle organ. For that, let's draw a large circle which would be the cross section of the muscle as an organ. Okay? So then label this as skeletal muscle, which is the organ. Like we have brain as an organ, like we have liver as an organ, like we have stomach as an organ, because they are made up of different type of tissues, right? Okay. Okay, then around this whole muscle, let's draw a thick layer of connective tissue. Dense, irregular connective tissue. What should we label this? Remember, the thick, dense, irregular connective tissue covering the whole muscle is called epimysium. Okay, so then here we are going to call this as epi means above, mysium means muscle. Okay, then inside this, let me use another color. Inside this big circle, let's draw a few smaller circles or bundles. Then these bundles are fascicles, group of muscle fibers. And also do not forget to label this as muscle fascicle, which is the bundle of muscle fiber. What should we do right now? We have to protect this structure as well, right? Okay, now let's draw another connective tissue around each muscle fascicle for protection, for support our muscle fascicle. So then, what should we label this? Now, this connective tissue which surround each muscle fascicle is called peri, means around, mysium, muscle. Okay, fine. Then let's go inside of each muscle fascicle. And I'm going to use another color this time. And then each of these would be our muscle cell, right? Or muscle fiber. Many muscle fibers. Then now I'm going to label this as muscle fiber or muscle cell. And do not forget, we have to protect our muscle cell or muscle fiber. Now we are going to draw another connective tissue. Let me use this color. Okay, so then around each muscle fiber, we are going to draw a layer of irregular connective tissue, which would be the innermost layer of the skeletal muscle sheath. And then now we are going to call this as endo, means inside, endomysium. Okay. So, but we are not done yet, because now we are going to look at one muscle cell and make it out and zoom in, so that would be our muscle fiber or muscle cell. So then you will see that each muscle cell actually is composed of many myofibril. So then each of these would be myofibril, which would be at the microscopic level. So if again we zoom in each myofibril is composed of myofilaments. Filament, okay, which is contractile protein. Tile protein. Okay, so you should be done with this part. And now let's take a look at the skeletal muscle fiber and know them better. So muscle fibers are special cells. They are special because actually they are the only cells in our body that they are able to contract and make movement. So skeletal muscles are called muscle fibers. Why? Because of their long and thread-like shape. You know, unlike most cells in the body, muscle fibers are very long and can run even the entire length of a muscle. They can be up to the 12 inches. So that's the reason we can call them as muscle fibers or muscle cells. Like other cells, we have to define their plasma membrane, cytoplasm. They have some organelles inside of the cell. They have a nucleus. So the plasma membrane of a muscle cell is called sarcolemma. So sarcolemma means the plasma membrane of a muscle fiber or muscle cell. The sarco is the prefix means muscle. For example, we have like terms like sarcolemma, sarcoplasm, sarcomere. So these are means they are related to the muscle. So next, the cytoplasm of muscle cell is called sarcoplasm. Again, sarco means muscle, right? You know, the muscle cells for contraction needs lots of energy. And guess which organelle should be a lot inside of the muscle cell? That is correct, the mitochondria. So they have many mitochondria to produce energy for contraction. And because they are so long, so they have many nuclei. So they are multinucleated cells. And these nuclei would be found along the edges. So we can call them as peripheral nuclei, as they would be found like along the like ridges of the cell or just under their plasma membrane, under their sarcolemma. Then, so as you can see, we have sarcolemma, the plasma membrane of the muscle cell. Then inside of their cytoplasm or sarcoplasm, they have many organelles like mitochondria for energy production. And we also learn about the number of nuclei they have and the location of the nuclei, which would be found just under the plasma membrane, the peripheral nuclei. Because they are special, so it makes sense to have some modified structures, which we cannot find them in any other cells, right? So these modified structures only going to be found in the muscle cells would be called myofibrils. So then if you look at the sarcoplasm or inside of their cytoplasm, it's full of the myofibrils. And then also there is sarcoplasmic reticulum. The organelle is very similar to the smooth endoplasmic reticulum in the regular cells, but in the skeletal muscle cells, they're going to be called as sarcoplasmic reticulum, and they have a special function as well. And also there is another modified structure is called T-tubule, which is unique for the muscle fiber and only going to be found in our skeletal muscle cell or skeletal muscle fiber. Let's take a look at each of these structures one by one, and let's start with the myofibril. So if you remember, inside each skeletal muscle fiber, we find myofibrils. So this picture is a picture of a portion of a skeletal muscle fiber or skeletal muscle cell. They have nuclei, we have the sarcolemma, sarcoplasm, and look, inside of these skeletal muscle fiber, these all dots would be our myofibril. If we zoom in and make it kind of bigger, then you will see that the myofibril is actually the long cylindrical structure that runs the entire length of a muscle fiber from one end to another end. Each muscle fiber, which is just one muscle cell, contains hundreds or even thousands of these myofibrils. In fact, myofibrils are so packed inside the cell that make up over 80% of the sarcoplasm. So then let's look at the myofibril. Each myofibril is made up of these myofilaments, which would be thin filament and thick filament. And also, if you look at each myofibril here, you will see that it's actually made up of repeating segments. These repeating segments are called sarcomere. Sarcomere, inside of the sarcomere, actually we have the thin and thick filaments. Each sarcomere is the basic unit of muscle contraction. So make sure you know the myofibril would be found inside of the sarcoplasm of a muscle cell, kind of occupy 80% of the sarcoplasm, and then each myofibril, which is made up of thin and thick filaments or myofilaments. If we look at each myofibril, we can see these repeating segments, which each segment is called sarcomere. Okay, let's review right now. We talked about the myofibril, which they are cylindrical and long structure. Their length is like the equal as the entire muscle cell. They are gonna occupy 80% of the sarcoplasm. Then we each myofibril contain myofilaments, which each myofilament is composed of thin and thick filaments. And also we talked about each myofibril is composed of the repeating segment, which is called sarcomere. Remember that sarcomere is a basic unit of contraction. What does that mean is a basic unit of muscle contraction? Because when a muscle contracts, these segments, these sarcomeres, shorten. And that's how your muscle generate movement. So that is the reason we call the sarcomere as the basic unit of contraction. And each sarcomere would be found between two zigzag lines or Z lines or Z discs. So then, as I mentioned, when the muscle contracts, they're pulling the Z disc closer together, so it means sarcomere shortens, and then we're gonna have the movement or contraction. And now it's time to look at the structure of a sarcomere. So as you can see, we started from this large structure, the muscle, and then get smaller and smaller and smaller. We talked about the muscle as an organ, the muscle fascicle, the muscle fiber or muscle cell, and right now we are inside of a muscle fiber or inside of the muscle cell. Inside of the muscle cell, we found lots of myofibril, and then let's get smaller. Each myofibril is composed of these repeating segments, which is called the sarcomere. And we know the sarcomere is a basic unit of muscle contraction. Let's take a closer look at the structure of the sarcomere and its different regions. These regions help us understand what gives a skeletal muscle its striped or striated appearance under a microscope and how muscles contract. Let's start with the start point and ending point of a sarcomere, which would be this Z line or Z disc. So we have Z line or Z disc. Here. So this is where the sarcomere begins and ends. So one sarcomere goes from Z disc to another Z disc. So then this region would be called as sarcomere. You can think of it like a fence that holds the ends of each sarcomere in place. When the muscle contracts, the Z discs move closer together. The Z disc actually is made up of some special proteins. Okay, and then we can actually find a lighter region that would be found on both sides of the Z disc. So from here till here. This lighter area, when you look under a microscope, it's called eye band. So we call this lighter region as eye band. Just remember the eye is a thin letter, then the eye band is made up of thin filaments. Thin leather, thin filaments. So here we have thin filaments, and that's the reason we can see this area under microscope as a lighter area. It's located on both sides of the Z disc, stretching into two neighboring sarcomeres. Okay, next. Next, we can define A band for the sarcomere. So next region of the sarcomere is called the A band, which the A band is a darker region. It would be found in the middle of the sarcomere. So from here till here, that would be our A band. A band contains the entire thick filament, including areas where thick filament and thin filaments overlap. So as you can see here, these red lines are representative of our thin filaments, but then these purple lines are representative of our thick filaments. So the entire length of the thick filament would be our A band, but then as you can see, in some areas we have overlap between thin filament and thick filament. All right, so then A band would be all filaments. So just remember, A band start with A, and all filaments start with A. So it means it is made up of thin and thick filament. Okay, next we have the H zone. Where is it? H zone here. H zone or H band is actually the lighter region which would be found at the center of the A band. So inside the A band, there's a lighter zone called the H zone or H band. This area has only thick filaments. So if you look at this area, you only can find these purple lines. There is no overlap between the thick filament and thin filament in this region. It's visible when the muscle is relaxed. But during a contraction, H zone disappears. Then we have another structure for the sarcomere, which is called M line. The M line would be found just at the center of the sarcomere. Inside the H zone is the M line. This is a line made up of proteins that hold the thick filaments in place so they stay lined up during contraction. Think of the M line as the anchor or center pole holding everything steady. So then we actually talked about the Z disc, I band, A band, H zone, and M line through this picture. Now let's review it one more time. We actually were finding the region of each sarcomere is made up of several region. Then we have Z disc or Z line. We have I band or lighter band. We have A band or darker band. We have H zone and M line. So for the Z disc, we said this is the beginning and end of the sarcomere. It consists of the proteins that would be the attachment site for our thin or actin filaments. Then we talked about the I band, which is the lighter region. We can see it under a microscope. It's only contain our thin actin filament. So I is a thin letter and it's made up of thin filament. Okay, and then we talked about the A band, which would be found at the center part of the sarcomere. It contains both or all filaments, like the thick filament and thin filament, myosin and actin filaments. And then it appears darker under a microscope because of presence of both types of filament, which makes sense. And then we talked about the H zone is important to know that. H zone is visible when sarcomere is relaxed. So this is going to be found on either side of the M line at the center of the A band, which is a lighter region. And the reason is lighter because here we only have the thick filament. There is no overlap between thick filament and thin filament in this region. And finally, M line would be made up of protein at the center of the sarcomeres, which hold the thick filament in place. And now it's time to draw a sarcomere and fully understand the structure of the sarcomere. Let's draw a sarcomere together, which we know the sarcomere is a basic unit of muscle contraction. Are you ready? Let's start by drawing a zigzag vertical line on left side of your paper. Okay, and the other side as well. This is Z line or Z disc. So this is not art class. Don't worry about like I'm a terrible painter. So that is you see, but it's important to draw it and you can learn it from that. So let's call this a Z disc or Z line. Both are correct. Okay, now mark or draw a bracket from one z-disc to another z-disc and label the entire section as one sarcomere. From each z-disc, draw about like 3 to 4 straight thin lines. Toward the center, but leave a gap in the middle. Okay, so they're not going to touch each other. So they're coming from the z-disc and then going toward the center. But as you can see, there is a gap between these thin lines. And what should we call this? Don't forget to label this as thin or actin filament. Now in the center between the thin filament, let's draw one thick line. This is your thick filament. Okay, and do not forget to label this as your thick or myosin filament. Then as you can see, we have some areas that they overlap with thin filament. Okay, next label the length of the thick filament. I'm choosing the color. Let's use this one, the blue one. Okay. So then label the entire length of the thick filament from here, the entire length of the thick filament from here till here as what? As the A band. So here we have our A band. So as you can see, our A band is composed of the entire length of the thick filament, and also we have some parts that they overlap with the thin filament. Then on either side of the sarcomere where there is only thin filament, okay, so if I want to show the thin filament at the other side of the sarcomere, should I do this? But here we can call this part as I band. So it means the part of the I band would be found in this sarcomere, and another part of the I band would be found at the adjacent sarcomere. So here, from here, we have our A band, which only composed of our thin filaments. Okay, then let's go in the very middle of the sarcomere, within the thick filament area, within the A band. Then here, as you can see, at this area, which only the thick filament would be found and there is no overlap between thin and thick filament. It would be a lighter zone in the middle of this A band. We call this as what? As H zone or H band. Then inside of the H zone, let's draw a line which would be our M line. This vertical line would be our M line, which anchors the thick filaments. So that is the structure of the sarcomere in a relaxed sarcomere, when the sarcomere is not in the contraction. So hopefully you learned about the sarcomere structure and the region. We have ZDs, we have I band, A band, H zone, M line, and then as you can see inside of the sarcomere, we have thin filament and thick filament, actin filament and myosin filament. As you remember, we are talking about the structure of the skeletal muscle, which is unique and only going to be found in our skeletal muscle cell. Let's talk about this unique structure, which is called transverse tubules or T-tubules. T-tubules are tiny tubes that are connected to the muscle cell membrane, which the muscle cell membrane or sarcolemma, then it goes deep into the muscle cell and make these little tunnels or tubes. So if you look at this picture, as you can see, that is our sarcolemma, right? The plasma membrane of the muscle cell. And then here we have the muscle cell membrane or sarcolemma, which invaginate into the muscle cell and make this tiny tunnel or tiny tube. Each is called transverse tubule or T-tubule. These are important because when your brain sends a message to your muscle to move, the message is an electrical signal which this gonna travel quickly deep inside the muscle, not just on the surface. This way the whole muscle cell contract at the same time. So then let's look at this regarding the transverse tubule and just review everything regarding the transverse tubule or T-tubule. So these are the tiny narrow tubes when the sarcolemma goes deep into the muscle cell would be called transverse tubule. So they extend into the sarcoplasm at the right angles to the cell surface, and these are actually would be the passageway through the muscle fiber. What is going to pass through the transverse tubules? The electrical signals. Right? Okay, so the electrical signals would move through these tiny tubes or transverse tubules. And we also talked about the function of transverse tubules, which they facilitate transmission of the action potential or those electrical signals into the muscle cell, because we want to make sure that these electrical signals reach to all parts of the muscle fiber at the surface and also in the deep of the muscle cell at the same time quickly. And then at the end, we are going to have the muscle, the whole muscle cell contract at the same time. Next structure of unique structure for muscle cell is called sarcoplasmic reticulum. The sarcoplasmic reticulum is kind of like smooth endoplasmic reticulum, which we learned about the organelles in the cell chapter. But the sarcoplasmic reticulum, it has a special job in muscle cells. It forms a tube-like network that wraps around each myofibril. And the main job of the sarcoplasmic reticulum in muscle cell is to store and release the calcium ions. Why calcium ions? Because calcium ion is very important for muscle contraction. Without calcium ion, the muscle contraction won't happen. So make sure you know the sarcoplasmic reticulum is an organelle, would be found in muscle cell, is responsible to store calcium ion and also release calcium ion, which is essential for muscle contraction. There is another unique structure for skeletal muscle cell, which is called triad. Let's learn about something called a triad in muscle fiber. A triad is made up of 3 parts. That's why it's called a triad. Means 3 structures. This triad is actually composed of 1 T-tubule. So number 1 is transverse tubule or T-tubule, and 2 terminal cisternae. 1, 2. 1 T-tubule and then 1, 2 terminal cisternae. So then a total would be 1, 2, 3, right? So this terminal cisternae would be found on each side of the T-tubule. We know about the T-tubule, right? It was a tiny tube or tiny tunnel that carries the electrical signal deep into the muscle cell, but terminal cisternae actually are the swollen areas of the sarcoplasmic reticulum that sits on both sides of the T-tubule. They store and release calcium, which is needed for muscle contraction. We talked about this before. So the terminal cisternae, just a part of the sarcoplasmic reticulum. So together, these triad helps the electrical signals from the brain quickly cause the release of calcium from the sarcoplasmic reticulum. And that's what starts the contraction. So then a triad is like a sandwich with a T-tubule in the middle and two calcium storage sacs on the side. So a combination of these, a pair of terminal cisternae, the part of the sarcoplasmic reticulum, plus a T-tubule, like a sandwich, is going to make the triad. And make sure you know the components of the triad and function for each of these components. Terminal cisternae is the swollen part of the sarcoplasmic reticulum on either side of the T-tubule, which when the signals coming from our nervous system, then it would release the calcium. Then T-tubule would kind of carry the electrical signals to the deep part of the cell. So then it helps like make sure the muscle gets the message to contract and does it fast. Plays a critical role in muscle contraction by coordinating the release of calcium ion. Now it's time to look at the structure of the thin filament. Thin filament is also called the actin filament. So thin filament or actin filament. Thin filament is made up of three main proteins. We have actin protein, tropomyosin, and troponin. The actin protein or actin molecule is a globular molecule. This actin molecule would join together like a monomer, join together and make the polymer, which this polymer is called F-actin. So each of these globular molecule is G-actin or globular actin, and then together they're gonna make F-actin. To make our thin filament, actually we need these two F-actin filaments twisted on each other. So this is a long twisted chain made up of many small units called G-actin molecule. So just imagine it like a twisted string of beads. Then each G-actin, actually it has the active site where the myosin molecule can attach. So that is our actin molecule, as we mentioned. And each actin molecule, it has this active site or binding site for myosin. Okay, so then let's talk about troponin. So troponin is another protein, which actually troponin, it has three small parts. So each troponin molecule has three parts, has three small parts. One part hold another protein called tropomyosin. So here you can see the tropomyosin. Another parts bind to the G-actin, and then another part binds to calcium ion. So one of these parts binds to the actin, another part binds to tropomyosin, and then another part would be bind to the calcium ion. Okay, so then when muscle needs to contract, we need calcium, and then calcium would be bind to the troponin molecule. Let's talk about a little bit regarding the tropomyosin. Tropomyosin is this long double-stranded protein that you can see wraps around the actin filaments. Actually, it's tropomyosin is going to cover this active site for myosin. When the muscle is relaxed, the tropomyosin would cover the active site of myosin on G-actin molecule. Its job is to block myosin from attaching when the muscle is relaxed. So that is the important protein molecules are making this thin filament. So one more time, let's review it through the text. So we are talking about the thin filament. We know it's made up of three main proteins: actin, troponin, and tropomyosin. Because actin is going to make the background for the thin filament, we also can call the thin filament as actin filament. And we know the thin filament is made up of two twisted strands of F-actin, which each one is composed of around 300 to 400 G-actin molecule. And we also said each G-actin molecule has an active site for myosin binding. And then we talked about the troponin, which has three parts. Each part would be bind to the different structures, like one gonna be bind to the tropomyosin, and its job is to kind of lock tropomyosin in place, and another segment would bind to the G-actin, and another part it has a receptor for calcium ion. Then tropomyosin, which is a double-stranded protein, is going to cover the G-actin's active site during the relaxed situation of the muscle. So this actually going to prevent the myosin bind to the actin or myosin interaction. Now let's talk about the structure of the thick filament. So the thick filament is contain only one type of protein, which is easy to understand. So thick filament is called a myosin filament. So it only has this main protein, it's called myosin. So each thick filament, it contains around 300 myosin molecule. For each myosin molecule, as you can see here, we can define a tail and two elongated globular head. So all of these tail stick together and form the thick filament. And as you can see, these heads, these two heads or round heads called myosin heads stick out. So these are the myosin heads sticking out, and these are the tail of the myosin molecule. So these myosin heads are super important because they attach to actin filament during the muscle contraction and pull on it to make the muscle shorten. So do you remember the active site for myosin? And here on the head of the myosin molecule, we can also find two attachment sites. One attachment site for ATP, because we need energy for muscle contraction and relaxation. And also one attachment site would be for actin molecule. So we have these two attachment sites. Attachment site on the myosin head, one for actin and another one for ATP. Now let's talk about how muscles actually contract. We are using this sliding filament theory to understand how muscle actually contract. During the contraction, our thin filaments slide past the thick filament. So as you can see in this picture, that is our sarcomere at rest. And this is the sarcomere contraction. And filament sliding occurred means the thin filament slide past the thick filaments. So for this to happen, the myosin heads must grab onto the actin filaments and pull them toward the center of the sarcomere. So then this pulling action shortens the sarcomere. So if you compare the length of the sarcomere between the sarcomere at rest and sarcomere contraction, you will see that the sarcomere length has been shortened, which makes the whole muscle fiber get shorter and creates a movement. So then we have the shortened sarcomere by muscle contraction because our thin filament slides over the thick filament. So we have more overlap between actin and myosin filament. That's the reason our eye band gets shorter, because now we have less area, we only have thin filament, but more area we have overlap. Because if you remember, our eye band, it would be the whole length of the myosin filament or thick filament. So the thick filament is fixed and it's not gonna change. So the eye band stays like the same length, but because we have more overlap between the actin and myosin in comparison with the sarcomere at rest, so then the area which we have our H band, it gets shorter or sometimes disappear. So what would be the result of the muscle contraction? So the I band gets shorter, the H band gets shorter or sometimes disappear, and the whole length of the sarcomere shorten. But the A band remains like unchanged, so it's not gonna change. No change at the length of the A band. Okay, let's see what do we have here. So we know that the sliding filament theory is actually would be when the thin filaments slide past the thick filaments. That is when the myosin heads bind to the actin and pull the actin to the middle of the sarcomere. And for this to happen, we need lots and lots of the ATP or energy, which the mitochondria are responsible to uh, produce the ATP. And these are the changes that are going to happen during the contraction. H bands and I bands get smaller because now we have more overlap between actin and myosin. That's the reason our I band, which is the only thin filament, gets smaller, and H band, which would be the only thick filament, also gets smaller, and the zones of overlap gets larger. So then, but the A band remains constant and it's not going to change. But because Z lines move closer together, the length of the sarcomere is also get smaller. Okay. Now let's see how this muscle contraction is going to happen from the beginning. You know, a skeletal muscle cell only contracts when it gets a signal from the nervous system. So we should have a signal from our nervous system, from a neuron, from a motor neuron, to send these signals to the skeletal muscle and say, hey, it's time to contract. So this signal happens at a special connection called the neuromuscular junction. So the neuromuscular junction is the junction or the synapse between a neuron and a muscle cell. So through this neuromuscular junction, these electrical signals can pass from a nervous system, from a neuron to the muscle cell. The neuromuscular junction, it has three main parts. We can define, so here you can see how the motor neuron reaching to the muscle cell, and now we have here the connection the contact between the neuron and muscle cell, which is called neuromuscular junction. Let's zoom in the neuromuscular junction and see what would be found at the neuromuscular junction. These are three parts. We have axon terminal, which the axon terminal or synaptic terminal is the end of motor neuron's nerve fiber that sends the signal. And then we have motor end plate, which the motor end plate would be actually the special part of our muscle cell membrane or the sarcolemma that receives these electrical signals from the neuron, and then there is a tiny space between the nerve ending and the muscle plasma membrane, or between the axon terminal and the motor end plate. This tiny space is called synaptic cleft. So then when the motor neuron sends a signal, it crosses the synaptic cleft and tells the muscle to contract. So the neuromuscular junction is like the communication bridge between the nerve and the muscle, making sure the muscle only moves when the brain wants it to. So here, let's see, when we're talking about the mechanism of a skeletal muscle contraction, the first thing that you have to remember is that we need this stimulation from a motor neuron, which the motor neuron is part of our nervous system. It's going to send the signals to the skeletal muscle cell, and then when the skeletal muscle fiber is going to meet the neuron, that site is called neuromuscular junction, or NMJ. The specialized site when the skeletal muscle fiber would be controlled by the nervous system. And also remember that the neuromuscular junction has three main parts: axon terminal, motor end plate, and synaptic cleft. The axon terminal is, or synaptic terminal is, the ending part of the motor neuron, and the motor end plate is a specialized region of the sarcolemma, the plasma membrane of the muscle cell, and a synaptic cleft, which is that tiny space between the neuron and the muscle. So make sure you know for the neuromuscular junction, which the name of this structure is descriptive, we have neuron or nerve cell. We have the muscle cell at other side, okay, and there is a space between them. So this space is called synaptic cleft. And then at the neuron side, we have axon terminal, the ending part of the neuron, and at the muscle side, we have motor end plate. Motor end plate. So do not forget, the motor end plate would be at the muscle cell side. Okay, let's see what is actually happening here. This is a bigger picture of the neuromuscular junction. We zoomed in here. That's, you can see the neuron side here. This is axon terminal. And this is a muscle side, which we have the motor end plate. So at the axon terminal, inside the end of motor neuron, which is called the axon terminal, we have tiny sacs filled with a chemical called acetylcholine. The acetylcholine is a chemical messenger, would be found in the sacs at the axon terminal. It's a neurotransmitter, is a chemical messenger that helps the nerve communicate with the muscle. This is how they just talk together. When the nerve sends the signals to contract, these sacs release the acetylcholine. Where? Into this small space, into the synaptic cleft. On the muscle side, on the motor end plate, we actually have the receptor for acetylcholine, So then we call this as acetylcholine receptors. So make sure you know the acetylcholine receptors would be found on the motor end plate, and then the vesicles or which contain the acetylcholine would be found on the axon terminal. So when the acetylcholine would be released into the synaptic cleft, then on the muscle side, we have acetylcholine receptors, which then they would bind to these receptors and cause the muscle to get the signals to contract. But after the message is sent, there's an enzyme, which this enzyme is called acetylcholine esterase. So we call this as acetylcholine esterase, which this acetylcholinesterase would be found here on this synaptic cleft. It quickly breaks down the acetylcholine so the signal stops. This helps the muscle relax and get ready for the next signal. So acetylcholine sends the contract message from nerve to muscle, and acetylcholinesterase helps clear that message away, and it's gonna end the muscle contraction. So here, let's take a look at the like these for mechanism of skeletal muscle contraction. We learned that the cytoplasm of axon terminal contains the vesicles, the vesicle full of the acetylcholine, which we know the acetylcholine is a neurotransmitter, is a chemical messenger, which the neuron gonna send to the muscle cell to change the permeability and other properties of that cell's plasma membrane. And then in the synaptic cleft also we said we have the enzyme, which this enzyme breaks down the acetylcholine, and the acetylcholine receptors would be found on motor end plate. So again, one more time, the acetylcholine vesicles would be found inside of the axon terminal. The acetylcholine esterase, the enzyme, would be found in the synaptic cleft, and acetylcholine receptors would be found on motor end plate. Okay, which then is leading to the muscle contraction. Okay, let's continue. Just remember, to begin a muscle contraction, the first thing we needed, it was the stimulation from the nervous system, which we saw that how the motor neuron gonna send this electrical signal to the muscle through the neuromuscular junction, right? Okay, and guess what gonna happen? Now our muscle cell, it gets the message. Then the electrical signals as action potential would be found at the surface of the cell membrane of the muscle cell. And remember, we had the T-tubule, those tiny tunnels, gonna be extension of the cell membrane, the sarcolemma, to the depths of the muscle cell. They're gonna pass these electrical signals to the depths of the muscle between the terminal cisternae, between the soilen part of the sarcoplasmic reticulum. Then they would be stimulated to release the calcium ions. Okay, so let's see what happened so far. We know when the electrical signals coming from the nervous system, the first thing we expect to release the acetylcholine in the neuromuscular junction. The acetylcholine has been released, and now the muscle gets the message, and the electrical signals can make the action potential to the surface of the membrane, the sarcolemma. Then through the T-tubule, this message, this action potential, can spread across the membrane and also along the T-tubules. Then when this sarcoplasmic reticulum at the either side of the T-tubule gets stimulated or excited, they would release the calcium. What's gonna happen after that? Now we have calcium found in sarcoplasm around these actin filaments. Do you remember what was the structure of the actin filament? It's made up of actin, it's made up of the troponin, it's made up of tropomyosin. And guess what would be the happiest molecule right now? the happiest molecule would be our troponin molecule. Why? Because troponin loves calcium and they would bind to the calcium ion. So then the calcium ion bind to troponin, and then because the troponin also bind to tropomyosin, binding calcium to troponin make the changes at the shape of troponin, then it's gonna pull the tropomyosin away from the active site on actin molecule. So they're going to expose the active sites on the thin filaments. Then myosin head bind to those active site and contraction cycle begins. So what would be the contraction cycle? So the contraction cycle is a series of interrelated steps that begins with the arrival of calcium ions. So we need calcium ions to start the contraction cycle. So the first thing that's gonna happen through the contraction cycle is exposing the active site and making the cross bridges. So the active site exposed cross bridge formation is happening at the start of the contraction cycle. So as I mentioned, the calcium ions bind to troponin. Troponin molecule then change the position, then it's gonna pull the tropomyosin away from the active site on actin. Then we have the myosin head, which they are fully excited and fully energized to bind to the actin molecule. Then they're gonna form the cross bridge. So what is the cross bridge? When the myosin head bind to the actin molecule, that is called cross bridge. And what does it mean, the active site exposed? The active site on actin molecule exposing because of the troponin bind to calcium ion and make changes of their shape and position. They're gonna pull away tropomyosin molecule. So if you look at this picture, you will see that how the myosin heads are here. Then when we have calcium, then calcium would bind to the troponin, and right now these active sites are naked, and then the myosin head would bind to the actin molecule. Okay, what's gonna happen next? So the next we have a myosin heads pivot. So after the cross bridge form, it means after myosin bind to the actin molecule, the energy that was stored in the resting state is released as the myosin heads pivot toward the M line. Means that the myosin, after the myosin heads attached to the actin, they pull the thin filament toward the center of the sarcomere. This movement is called Paver stroke. So look at this picture. So you see that here we have the head of myosin bind to the actin. They pivot and pull the myosin toward the center of the sarcomere, and this movement is called Paver stroke. And then when the Paver stroke happens, the energy stored in the myosin head is released. Okay, so then the energy would be released, and then the myosin releases the molecule called ADP and phosphate group. So at this stage, the ADP and phosphate group would be released. These pulling actions shorten the sarcomere, which causes the muscle to contract. Let's see what's gonna happen after the Paver stroke. So after the Paver stroke, a new molecule called ATP binds to the myosin head. And when ATP attaches, the connection between the myosin head and the actin's active sites break. So the myosin heads lets go. So we call this step as cross bridge detach. So When this is going to happen when we have the new ATP molecules bind to the myosin head. So as we said, the link between the myosin head and the active site on the actin molecule is broken. The active site is now exposed and able to form another cross bridge. So then what's going to happen next? Guess what? Right now we have the active site on actin on free. They are ready to bind to another myosin head and start the cycle again. So after the myosin heads lets go of the actin, it's break down the ATP molecule into ADP and phosphate. The energy released from this reaction is used to recock or reset the myosin head, kind of like pulling back a spring on a toy. Once the myosin head is ready again, it can bind to actin and repeat the contraction cycle. So this cycle happens many times every second as long as there's enough calcium and ATP available. So the myosin reactivates when the free myosin head splits ATP into the ADP and phosphates. So then this energy released is used to just reset the myosin head, recock the myosin head. Okay, so then we know as long as we have calcium, as long as we have ATP available, this cycle happens and happens and happens and happens. So let's review the key steps of contraction cycle one more time, starting when the myosin head attached to actin. So we have the myosin cross bridge attachment. Next step, we have power stroke. And then we said we have the cross bridge attachment. And then we have the myosin head reposition. And then again, they're gonna repeat several times or many times. So the first step, the myosin head attaches to the active site on actin. So here we have cross bridge formation. So when we have cross bridge formation, actually our myosin head is full of energy because it's super excited and it has a many energy, right? Okay, what's gonna happen next? So with this energy says, hey, I can move you to the center of the sarcomere. The myosin head pivots toward the center of the sarcomere. This movement is called power stroke. So we have power stroke. Then power stroke is pulling the actin filament inward. During these movements, we have these molecules ADP and phosphate group would be released. Then myosin, it gets tired, right? It gets tired and says, hey, you know what? I need new energy. So then at this time, new ATP molecule binds to the myosin head. Then it says, hey, I need a little bit rest. So that's the reason detachment is going to happen. The new ATP molecules bind to the myosin head and causing the myosin to detach. So we have cross bridge detachment. Okay, cross bridge attachment. So then the myosin head, then when it has ATP, says, hey, now I can do function. Let's break down ATP into the ADP and PI, and it gets more energy one more time. It's gonna use this energy to reset into the starting position. And then it's gonna like bind to another actin molecule. Again, this cycle repeats as long as calcium is present, ATP is available, the allowing muscle to contract smoothly. Okay, so then what gonna happen after that? We have muscle relaxation as well. Just do not forget the acetylcholinesterase enzyme. The acetylcholine is break down by the acetylcholinesterase, which would be found in the synaptic cleft. So it's gonna end the action potential generation. Then what's going to happen after that? The calcium ions would be reabsorbed. Remember, the sarcoplasmic reticulum, they can store the calcium. So then they're going to reabsorb the calcium ions, and then when there is no calcium ion, without calcium ion, tropomyosin return to their normal position, and again they're going to cover active site of actin molecule, and then without cross bridge formation, without those exposed sites, without calcium, so then the cross bridge not going to form, and then contraction ends. Then we have muscle relaxation occurs. So then, as you can see, for muscle relaxation, we need the acetylcholinesterase, the enzyme. We need to reabsorb calcium ions, return them back to the sarcoplasmic reticulum. Then the tropomyosin, troponin, they need to get back to their normal position. Now all of the active site on actin molecule has been covered, and then there is no way that the myosin head can bind to the actin, and this is the end of the contraction. Okay, and now let's go over some important terms regarding the muscle. We have muscle atrophy. The muscle atrophy happens when a muscle is not used or regularly stimulated by a motor neuron. When this happens, the muscle fibers get smaller and weaker. So the muscle atrophy means when the muscle fibers become smaller and weaker. This means the muscle loses their strength, endurance, so it cannot work as well. You might see this after someone has a cast on their arm or leg for a long time because of a fracture. This muscle isn't being used much, right? But the good news is that if the muscle use is only if the muscle use is only temporarily reduced, this atrophy can be reversed with exercise and physical therapy. We have another term is called muscle hypertrophy. Muscle hypertrophy means when muscle gets bigger and stronger. This happens when muscles are used regularly and intensely, like during weightlifting or other strength training exercises. Because the muscle works harder, the individual muscle fibers increase in size. So we have increasing the size of muscle cell. When we say hypertrophy for skeletal muscle is happening, it does not mean that we are increasing the number of muscle cell. It does not mean increasing the number of muscle cells. It means we are increasing the size of muscle cell. So our muscle cell get bigger. And that's because they gonna add more proteins inside of the muscle cell and getting thicker. So muscle hypertrophy means your muscle cells grow bigger, the muscle cells sizes get larger, bigger because of adding the proteins inside of the muscle cell. So one of the things that we can do for muscle hypertrophy is when you are going to do exercises, working out, and also having a protein diet, is actually helping the muscle cells to get bigger and stronger. And at the end, let's see what would be the effects of aging on skeletal muscle. As we get older, our skeletal muscle go through some important changes. So the muscle fibers become smaller in diameter, which means the muscle becomes thinner. The muscle become less like elastic. The more fibrous tissue build up. This is called fibrosis, making muscle stiffer and less flexible. Because of these changes, older muscle don't handle exercise as well. People may get tired more quickly and have less endurance. Muscles also don't regulate body temperature as well. Okay, so then and the ability to recover from injury also decreases. So in simple words, with aging, muscle gets smaller, stiffer, weaker, making it harder to exercise and recover from injuries. And at the end, let's test your understanding and see what you learned so far. Test yourself. The connective tissue covering a skeletal muscle listed from superficial to deep. Which one is that? So if you remember, we mentioned the connective tissue gonna cover skeletal muscle. Then we have the whole skeletal muscle, the muscle fascicle, and muscle fiber. And the um, connective tissue covering the whole muscle, it starts with epi. The connective tissue covering each muscle fascicle start with peri, means around, and this connective tissue covering each muscle fiber starting with endo. So mesio means muscle. So from superficial to deep, we should have epimysium, perimysium, and endomysium. If you choose this answer, you are absolutely correct. Next, question asking, between a resting muscle fiber, where is the greatest contraction of calcium found? In resting skeletal muscle fiber. We know calcium is important ion for muscle contraction, right? But when the skeletal muscle is resting, do we need calcium inside the muscle cell? The answer is no, right? So they should be stored, should be found in their storage. What is the storage of calcium inside of the skeletal muscle fiber? That is the sarcoplasmic reticulum. Sarcolemma is the plasma membrane. We do not have calcium ion in the plasma membrane, right? Tubules also are the extension of the plasma membrane into the muscle cell that's going to be carrying the electrical signals, not calcium. Sarcoplasm is the cytoplasm of the muscle fiber. So when it's asking within a resting skeletal muscle fiber, the calcium ion would be found a lot in the sarcoplasmic reticulum. But if it's asking when a muscle in the contracted situation, then we should expect to have more calcium ion in sarcoplasm because they should be released into the cytoplasm from the sarcoplasmic reticulum, right? Hopefully it makes sense. Vesicles filled with acetylcholine are found where? So if you remember, that is actually asking you about neuromuscular junction. You should know that the components of the neuromuscular junction, we have the axon terminal and the neuron end. We have motor end plate at the muscle and we have synaptic cleft between these transverse tubules, not even part of the neuromuscular junction. Let's cross this out, right? And think about these three. The acetylcholine is a chemical messengers which actually would be found inside of the vesicles in the axon terminal, right? But if asking where the acetylcholine receptors would be found, the answer would be the motor end plate. So here we have the acetylcholine receptors. And also do not forget the acetylcholine esterase enzyme would be found here at the synaptic cleft. And then we have one more question. Which molecule supplies the energy for muscle contraction? Remember, for muscle contraction, we need the ATP as the molecule which the mitochondria would produce the ATP, and for muscle contraction we need lots and lots and lots of the ATP. Thank you so much for watching this video. Feel free to comment under this video if you have any question, and good luck.